Labs 4 and 5 this week taught us techniques that allowed us to estimate the number of bacterial cells per mil in a culture. The techniques learned were serial dilutions, spectrophotometry, spot plating, and colony counting. The absorbance of a bacterial culture, read with a spectrophotometer, is directly proportional to the number of cells per mil in the bacterial culture. If the number of cells per mil doubled, it would be expected that the absorbance value of the sample would also double. Thus, absorbance can be used as an indirect means to observe bacterial growth in a population. A direct method to determine bacterial population in a culture involves a series of dilutions aimed at decreasing the concentration of bacteria to countable numbers. The dilution are prepared by consecutive transfers of a volume of bacterial culture into dilution tubes, using broth as the diluent. After the dilutions, a small volume of the diluted sample is plated. If the concentration of bacterial cells in the volume plated is low enough, each cell will grow in discrete areas of the plate, producing one colony. Each colony represents one cell present in the volume plated. So, one colony represents one cell or one colony forming unit in the volume plated. The number of cells per mil in the dilution tube can then be determined. So if 10 microliters of the 10 to the minus 4 dilution contains 11 CFUs, then in the 10 to the minus 4 dilution tube, there are 1,100 CFUs per mil, because we know that 10 microliters equals 0.01 mils. By multiplying the number of cells in the volume plated by the dilution factor, we can then calculate the number of CFUs per mil in the original bacterial culture. So, if the 10 to the minus 4 dilution tube has 1,100 CFUs, we have to multiply by the dilution factor to obtain the number of CFUs per mil of the original culture. So the original culture would have 11 million CFUs per mil. During our exercise, a 1 to 10 dilution of the original culture is created, and the absorbance value of that dilution is recorded. Then, a series of 10-fold serial dilutions are made and plated. Low dilution tubes may not give us countable numbers of colonies. On the other hand, high dilution tubes may not give us enough colonies for an accurate count. Using the dilution tube that give us an appropriate number of colonies, the CFUs per mil of the culture at time 0 can be calculated. Now we have recorded the absorbance of a 1 to 10 dilution of the culture that corresponds to a calculated number of CFUs per mil. After time 0, every 30-minute interval, a 1 to 10 dilution of the original sample will be created, and the absorbance value of the sample will be read. At the 1 hour mark, the absorbance of the 1 to 10 dilution is also recorded, and a series of dilutions are created and plated that would allow us to calculate the CFUs per mil of the culture at the 1 hour mark. Now we have the number of CFUs per mil after 1 hour incubation and the corresponding absorbance value of those CFUs per mil at the 1 hour mark. With the data collected of absorbance values every 30 minutes and the calculated titer every hour, 2 graphs will be created. The first graph will depict absorbance values versus time. The graph can be used to determine the generation time of the bacterial population. Generation time is the time that it takes the bacterial population to double in numbers. When the population reaches the exponential phase of the growth curve, generation time becomes constant. Knowing that absorbance value directly correlates with bacterial population, taking 2 absorbance values that double and finding the time that it took for those 2 absorbance values to double, we will find the generation time of the population. Similarly, graph 2 will be constructed by taking the calculated titer versus time. Generation time can also be determined by finding 2 points at which the number of the population doubles and determining the time intervals at which each of the doubles take place. These will give us the generation time by using the direct calculated titer.